Okay, so our conversation tonight is going to move into the next part of our cardiovascular chapter, which focuses on the blood. And so last time when we met face to face, we really talked about the heart, the chambers of the heart, the valves that we have involved with that process, and how that all kind of works together to move blood forward. And that's really the number one goal of those structures is to make sure that blood is always moving in one direction and one direction only. So for this particular conversation, we're going to now be looking at the blood itself and how it serves the body as it travels from the heart through all of the blood vessels and then back to the heart again. So when we look at blood itself, it is considered to be a connective tissue. And this is something that we talked about back in first semester. And the reason that it is considered to be connective tissue, even though we typically think of connective tissues as being very strong materials, you've got tendons, you've got ligaments, you've got cartilage that would be in that group. And now all of a sudden we throw this liquid type component in there. And what's the connection? The connection is going back to the two things that we talked about in that number one, the cells are what we call a matrix type design. So the actual cells themselves are separated by some manner and create a matrix. And because it meets that criteria, it can be considered a connective tissue. The other reason is the same cells that give rise to our cartilage and our tendons and ligaments in embryological development are the same cells that ultimately give rise to our blood cells. And for those two reasons, it is considered to be a connective tissue. Now, if you look at the packet that you have, it's going to start out with a slide that talks about the cardiac cycle. And this was something that I touched on very briefly in our last conversation. We talked about the SA nodes and the AV nodes and how that electrical impulse is going to travel from the brain down toward the heart. And because of the positioning of those nodes, it's going to hit that top one first, send the electrical current through the top two chambers of the heart, and get those two chambers to contract. As that electrical current continues to move down, it's going to hit that second node, which is positioned just a little bit lower. And that time differential for that travel to move from the top to the bottom node is just enough time for that top contraction to happen, get the blood filled into those bottom chambers so that when it hits that second node and the bottom chambers contract, now it's forcing that blood forward and it happens in a very rhythmic manner. So looking at the cardiac cycle packet that you guys have, I just want to continue on with that a little bit more because there's a lot more information that comes with that. When we look at the cardiac cycle, essentially we're looking at one heartbeat. And if you guys have ever seen an actual heartbeat, typically we think of flat lines as being a bad thing, but there should be a little bit of a flat line between the heartbeats themselves. That is okay. And when you have that kind of stereotypical image of a heartbeat on a monitor, you should have a small little peak, and then you should have a very high peak, and then you should have a dip, and then you should have another little peak at the end. So there's actually 4 different parts that you have to that particular heartbeat. And we're going to talk through what each one of those peaks and valleys, so to speak, is really telling you when you look at a heartbeat, so that if you get to see them quite a bit, just by looking at what those peaks and those valleys are doing, you can tell what is going on in the heart. So it's broken down into waves, and I told you you have a first little spike in a heartbeat. And if you look at your packet, that is referred to as the P wave. And in a P wave, the definition of the P wave, it talks about the contraction of the atria. And really what is happening is the nerves in that part of the heart are becoming depolarized. And again, this goes back to conversations that we had in first semester. So we're continuing to build on that information that we covered in first semester. In your nerve cells, your nerves are going to have positive and negative charges strategically placed, and they are kept separate on purpose. So until you actually want that nerve to fire an impulse, You keep the positive and the negative charges separated from one another, and we use the cell membrane to do that. So we place the positive charges on one side of the cell membrane, we place the negative charges on the other side, and we keep the cell membrane closed. And as long as that cell membrane is closed, no nerve impulse is going to fire. But when this electrical current comes into the heart, now it's going to tell that cell membrane, okay, we need those positive and those negative charges to interact with each other so that we can get this contraction to happen. Now, depolarization sounds like a really big word, but all that is happening is if you think about that setup, having positive charges on the outside, having negative charges on the inside, you created a magnet. Okay? If you think about magnets on your refrigerator, one side is positive, one side is negative. Now, if that particular setup, going back to our chem chapter, is called a polar molecule. Anytime you have a situation where something in the human body or some element is behaving like a magnet with one positive side and one negative side, that is polar. So if we're going to depolarize something, all that means is we're going to let those positive and those negatives interact with each other. So the channels on that cell membrane will open up, and it will cause those positive and those negatives to interact. Now, what direction is it going to go? So the setup of your nerves is you actually have these very large negatively charged molecules on the inside of the nerve cell. And because they are so much larger, they are going to pull those positives into the nerve cell itself. So the draw is going to be when those gates open up on the cell membrane, that those positives get pulled in. And as those positives come in and start to cancel out those negative charges, so one positive to one negative, they cancel each other out. As that happens throughout the entire nerve cell, that is what we call depolarizing it. So it sounds like a really big word. It's just talking about the movement of those ions and canceling each other out. The flow of those positive and those negatives moving are going to continue that electrical impulse through the cells that you're dealing with. In this particular case, we're dealing with this happening in the heart. So that P wave, that first little spike that you are going to get in your heartbeat reading is going to show the atria depolarizing. That is telling you that those negative and those positive charges are moving where they're supposed to, and that contraction of those top two chambers of the heart is going off without a hitch, no problem. So you get this tiny little spike, and then the next spike that you would see on your heartbeat is going to be what we call the QRS wave, also referred to as the QRS complex. Now, when you look at this is going to be a very tall spike on the heartbeat reading. And what's happening in this particular case is it refers to the contraction of the ventricles, but it's the same thing happening in the ventricles. It's those nerve cells depolarizing. So now we're seeing those positive and those negative charges happening within the cells on the ventricle side of the heart. And when that happens, it's going to give you that very tall spike. Now, the atria give you a tiny little spike, and then we get this huge spike from the ventricles. And if you think back to our conversation last week, there's an explanation for why you would have the same process happening, depolarization in the atria, depolarization in the ventricles. Why do we get such a difference in what those readings look like? Small spike for the atria, big spike for the ventricles. Goes back to what I told you about the muscle walls. Because the muscles are thin around the atria, even when they contract with a smaller amount of force, and so you just get a little spike. With the ventricles, because those muscles are so much thicker, you get a very pronounced spike because of the force that those muscles can contract with. Now, after those top two chambers contract, the bottom two chambers contract, and we had all of those positive and negatives moving around, there has to be a time and a place for everything to kind of recover. And so I told you that after the QRS wave, you're going to see a slight dip. And that's the process of the heart starting the refractory period, which means everything is starting to realign and get back to its starting point, so to speak, so that the next heart rate or heartbeat, can happen. Which leads us to the final little spike that we have, which is the T wave. And the T wave is when the chambers of the heart are repolarizing. So we talked about polar, positive on one side, negative on one side, acting like a magnet. Depolarization is when the positive and the negative start canceling each other out. Repolarization simply means that the positives are going back to where they need to be, the negatives are getting back to where they need to be, and the cell membrane is going to close to keep them separated again. So that process of everybody repositioning to where they need to be is what happens during repolarization. So if everything is happening the way that it should, you will get very definite small spike for the atria, big spike for the ventricles, a little dip telling you that everything is starting to reset, and then that last one, the T wave, where all of the positive and negatives are in fact getting back to where they need to be for the next heart rate or heartbeat to take place. So that is the information that is being provided through those heartbeat readings. The next slide, the electrical impulse, you can skip over that one. You find your slide that looks at the cardiac abnormalities. And there are so many of them out there, and I don't expect you to commit them to memory, but just to kind of look at a couple that you would see. Anytime that a heartbeat is not taking place or happening the way that it should, we call that an arrhythmia. And on the cardiac abnormality page, it just shows you 3 of the common arrhythmias that are out there. So the first one would be ventricular fibrillation, and it gives you the definition of just having a rapid and uncoordinated depolarization of the ventricles. So that's telling you that when it's time for those ventricles to contract, they're not doing it the way that they are supposed to. It's happening so fast and so quick that nothing really has a chance to reset and get back to its starting spot. So you'll notice you don't have any tall spikes in that first reading. And that's because those positives and those negatives never get reset. And so those muscles around the ventricles never get a chance to contract with the full amount of force that they should. So you're not going to see that in that particular reading. Tachycardia is simply when you have a rapid heartbeat. And the defining feature with a tachycardia is that you don't have any flat line. You should see flat line between each heartbeat if somebody is having a normal rhythm. And in this particular case, you'll notice that it is just rapid fire, one heartbeat after another. You see the tall spike for the QRS wave, not a really good P wave in there or not a really good T wave in there. You do see that little dip that I talked about, but that's just simply because they're happening so fast that you, again, don't get that good rhythm or that good reading. And then the atrial flutter, which is on the bottom, basically what you have happening there, you see kind of the waves and then you get a tall spike every once in a while. With those wave-like sections, it refers to the rapid atrial depolarization. So again, they're not ever getting a chance to really reset so they can do a good full fire or full contraction. And so instead you just get those little waves, and then every once in a while you still see that QRS. And because you get that tall spike, that tells you, okay, the ventricle side is doing what it's supposed to, and the problem with the reading is happening with the atria. So you can pinpoint where in the heart the problem is actually coming from. Like I said, there are tons of different arrhythmias and different types of readings that you could get. Those are just 3 of the more common ones that you would see on a day-to-day basis. So with the next set of slides that we have, it does start to get into the blood itself. And I kind of led off with that a little bit earlier, talked about it being a connective tissue. But one of the biggest things that you need to understand about blood is that it's really a transport system. It is giving a ride to oxygen and getting it delivered to where it needs to be in the body. So when you guys inhale and you take that oxygen into your system, we don't care that it got into your lungs so much. At that point, we need to continue to move it forward and get it to where it actually needs to be put to use, which is not even at the cell level, but all the way down into the mitochondria that you guys have in your cells. That is where you're actually going to be able to take that oxygen, put it to work for the body, and build ATP so that you guys have the energy you need to continue to work. So blood is a transport system to make sure that once you inhale that oxygen, you have a means to then pick it up and transport it and ultimately drop it off at every single living cell that you have throughout your entire body. One of the other things that your blood will do for you is it does help to distribute heat through your system so that you can maintain your body temperature. Obviously, that's a very important feature. The heat, a lot of the heat that you guys have for your body to utilize actually comes from your muscles. So one of the waste products of your muscles is that they will give off heat through the work that they do. And then that is transferred into your blood, which will help circulate it and make sure that all of your other tissues are staying warm and at that 98.6 degrees that they need to maintain. The components that you guys have in your blood, the red blood cells, white blood cells, and your platelets, we're going to talk about those a little bit more later, so we will then move on to blood volume and composition. So when you look at your blood volume, it talks about it being about 8% of your weight. Obviously, male, female, compared to kids as well, the amount of blood volume that you have in your body does vary somewhat. And so that 8% will be different from one person to the next. 55% of your blood is going to be a plasma. And plasma is going to be a material, I don't know if any of you have donated plasma, but they'll actually take your blood, they'll centrifuge it, and through that centrifuge process, they will separate your red blood cells from the plasma. Plasma is a liquid that you can see through, has a yellowish kind of amber tint to it. And then they can, once they centrifuge it, they can take those red blood cells and put those back in you. But that yellow liquid is going to be the plasma portion and accounts for just over half of your blood makeup. The remaining 45% of your blood is then going to be your actual blood cells. And so you are looking at your red blood cells. Red blood cells are going to be the ones that deliver oxygen. They have a special component to them known as hemoglobin. And hemoglobin are going to be the ones that pick up oxygen and give them a ride to where they need to be. Then you have your white blood cells, and those are going to be the defenders of your body. So they patrol your system, and anytime they come in contact with something or find something that is floating around in your body that's not supposed to be there, they will attack it and try to get rid of it. Then you also have your platelets, and your platelets are going to be fragmented cells, and we'll talk about those a little bit more, like I said, later. If you flip over to the red blood cell characteristics, so when we talk about your red blood cells, the proper name is erythrocyte. Erythro is a prefix that actually means red. Cyte, we've talked about many times, means cell. So literally the name means red cell. And when we talk about the red blood cell, it actually has a lot of really unique characteristics. It's referred to as being biconcave. So it's telling you that both sides are going to have a concave type design to them. And if you think about it, it kind of looks like somebody took it and pinched it in the middle. It's not a donut where it's hollow through the middle or there's a hole through the middle. It just thins out through the center. So it's thicker around the edges and then it thins through the middle. And the reason that that is so important, nothing in the human body happens by chance. I know you guys are tired of hearing me say that, but it is very, very true. So if we take this blood cell and all of a sudden we don't make it this nice round cell, but we give it a dip on both sides, what could be the potential reason for that? Well, think about it. If you have a straight line and you were to measure that versus a dipped line and measure that, which one's going to be longer? Obviously, the line that dips down is going to ultimately be longer if you were to measure it compared to that straight line. So by taking that cell and giving it those two curved sides that dip in, what you've actually done is you've increased the surface area. Why is that so important to our red blood cells? You can really make it more efficient by increasing that surface area, because if we give it more surface area, it now can pack in more hemoglobin, which means it can give more oxygen, a ride to your cells. You guys are trillions of cells in size. So if we can do any little thing to increase the efficiency of those red blood cells to be able to deliver oxygen to your cells, obviously we're going to take advantage of it. And that's exactly what the red blood cells did. Now, in order to do this, your red blood cells start out like a normal cell. They have a nucleus and they will go through their maturation process to a certain point. And once they get to a certain point, they do something that we really don't see in any of our other cells. It kicks out two of the largest organelles. Now, the largest organelle in any cell is typically your nucleus. So it kicks out that nucleus, which at that point, when you get rid of the nucleus, you get rid of all the instructions on how to divide. So once that nucleus is gone, your red blood cells can't divide anymore. The other thing that will happen is it will kick out the mitochondria. Mitochondria are not large by themselves, but because you have a massive number of mitochondria in your red blood cells, when you kick them out collectively, now you've freed up a lot of space. Again, why are we doing this? Well, if we kick out the two organelles that are taking up the most space in the red blood cell, again, we have provided a lot more room to pack in hemoglobin and make the red blood cell even more efficient at getting oxygen packed in and being able to deliver it to your cells. So your average red blood cell, if you look at it, it talks about it lasting about 120 days. And your red blood cells are moving in and out of blood vessels, bumping into each other. And so just wear and tear over time, after about 120 days, they are going to die and be flushed from your body. Typically, how you are going to remove those dead red blood cells is through your solid waste. And that really accounts for the coloration that you typically see in your solid waste, um, accompanied with bile that you have in your digestive system. But that brown color comes from those dead red blood cells. Now, the amazing thing about the body is I keep talking about the trillions of cells that we have, and we know that's a massive number, but sometimes it's really hard to wrap your head around just how big that number is. So if you kind of put things in perspective, you will lose 1,000,000 red blood cells every single second. 1,000,000 red blood cells every single second are dying off in your body, and you don't notice it. You don't feel the effects of it. Number one, you're replacing them as quickly as you are losing them, so you really shouldn't feel the effects of it. But to be able to lose 1,000,000 of anything every single second and still be able to function, that's pretty amazing that your body is able to keep up that cycle and still take care of everything else that has to happen within the human body. So just a massive number of red blood cells that are constantly getting replaced just because of the wear and tear of their movement, like I said, through the body. And we talked about the hemoglobin, and the hemoglobin, just understand, is really going to be responsible for making sure that oxygen is getting transported through the body and getting delivered to all of the cells. The next slide that you guys have talks about red blood cell counts, and I already mentioned this a little bit. When you look at males to females, there is going to be a slight difference. Generally, males are a little bit taller than females, and obviously children, depending on where they're at in the developmental phase, are going to be a little bit different. Your average male is going to carry somewhere between 5 and 6 liters of blood. Women are going to be closer to, 5 in that neighborhood, and children, it's really hard to put a scale on it because it just depends on if you're talking about a 1-year-old versus if you're talking about a 10-year-old. So 5 to 6 liters of blood when you're looking at your adults is what they typically carry. So if you think about if you have a 2 liter of soda, and if you were to put those on the table, how many it would take. So 3 of those 2 liters, and you're looking at the blood volume that you would have for the average man. Now on the slide, it actually talks about the number of red blood cells that you have in a cubic millimeter of blood, and it gives you those numbers. The reason that I wanted to put this particular slide on there is because there's a physiological difference between males and females as far as their physical ability, and this slide explains it. If you think about track records, so if you take an adult male and adult female and you put them on a track and you have them run 100 meters, 100-yard dash as fast as they can, or if you put 2 of them against each other and have them run a marathon, and then you look at the times for that. Typically, your males are going to have better times than your females. And that's not a slam on females. It has everything to do with what you're looking at on this slide. You have more red blood cells in a cubic millimeter of blood when you look at a male compared to when you look at a female. So if you start to build this out, okay, we've got more red blood cells in the same amount of space. If we've got more red blood cells, that means that their ability to deliver oxygen to their cells is better because they have more red blood cells in that space. So they are getting more oxygen delivered to their cells at a faster pace. If we're getting that oxygen to our cells faster or more efficiently, what does that ultimately mean? It means that your body is able to replace ATP better. So it would make sense that they are going to be able to run faster because as they are using up that ATP to run as fast as they can, it's getting replaced faster. So their ability to sustain that is going to be much better than that of a female. And again, it's like I said, it's not a gender slam whatsoever. It is very much a physiological process or explanation as to why you are going to see males run faster and be able to do things with better duration than a female. It has to do with their oxygen getting delivered to their cells and their ability to recover ATP at a faster rate because of it. If you look at the slide that talks about other components of the blood, you have the white blood cells, and your white blood cells get a proper name of leukocytes. And so if you break that down, you have the leuco, and then you have the sites. We know that site is always going to indicate cell. So anytime that you see that, that is your proper term or your proper way to refer to the white blood cells or the defenders of your body. I already mentioned that they are going to be the ones that patrol, and they do a variety of different things. Some of your white blood cells simply find things that don't belong there, but they don't do anything to destroy them. You have others that will attack and kill them. You have another group that is going to learn everything it can about those foreign bodies so that if they ever encounter them again, they know how to fight them off and deal with them at that point at a much faster rate. So a lot of different types of white blood cells that you have floating through your system. And we will spend a lot more time in our lymphatic chapter going through those in greater detail. So I listed the 5 major types of white blood cells that you have there. Neutrophils, you have your eosinophils, you have your basophils, your monocytes, your lymphocytes. If you pay attention to commercials right now, they have one out that talks about your reaction to certain things, and it's because of your eosinophils being overactive. So these are things that are coming out and we're seeing on TV a little bit more than we did in the past. You also have your blood platelets. Now, blood platelets start out as normal round cells. They actually come from a cell that is known as a megakaryocyte. Mega telling you that it's big, karyo indicating nucleus, cyte cell. So it's a very large nucleated cell in the beginning. What happens is when these megakaryocytes die, they don't just kind of disintegrate and go away. The best example I can give you, or the best visual, is to think of a glass plate. And if you were to take a glass plate and drop it on the floor, what would happen? Well, it's going to shatter, and it's not going to break into these nice even pieces. You're going to get these jagged edge shards of glass. And that's essentially what your platelets are. They are these broken, fragmented pieces from this megakaryocyte. And those jagged edges are very, very important because when you have an injury and you bleed, through the process of bleeding, you will begin to clot so that you can start the repair process. And what happens is you have these special little fiber threads that will create a net over the opening of the wound. And it's microscopic level. You can't see it, but it's happening. So you have the open wound. It indicates to your body that you need to start creating these threads, and they network across the opening of the wound. And what happens then is as you are bleeding, these platelets are coming out as well. But because they have those sharp, jagged edges, they get caught up in the net. And as you continue to bleed, more and more of those platelets will get caught in the net and they pack onto each other. And over time, they pack in there so tightly that they will stop the bleeding. It dries, it becomes the scab, and it will stay in there until the body has had a chance to repair. And then as new cells push up, they will push that scab off. The scab will eventually fall off the body and the repair process has happened. All of us at some point have gotten irritated with the scab and we pull it off before it's ready to come off, and what happens? You start to bleed again because you essentially pulled out the plug that your body had created to stop the bleeding at that location. Usually not as much as the initial injury, and the body will start it again, creating that network. Platelets get packed in there, stop the blood flow, and you get a new scab. So the last portion of this is going over blood types. And the blood types, I pulled some cards to show you and kind of give you some visuals, because when we look at blood types, there are 4 different types of blood types that we have in the human body: A, B, O, and AB. O is the most common blood type that we have. AB is the rarest blood type that we have. But getting an understanding of blood types and blood transfusions and why some work and some don't is what we're trying to accomplish with this last portion of our conversation. So when we look at the different blood types that are out there, so this first card that I have is showing you the blood cell itself. So you have the blood cell over here. And if you look at the surface of the cell, there's nothing on the surface of our type O blood cell. But if you look at off to the side, it has these different shaped structures. And on these round ones, it has the letter B. And in the triangle shaped ones, it has the letter A. So these are called the antibodies. You'll notice that the antibodies are not attached to the blood cell itself. So they are free floating, they are in the liquid portion of the blood, but they are not physically attached to the blood cell at any point in time. So for our type O blood, we have nothing on the surface. Those are called antigens. If you have anything stuck to the surface of the blood cell, it's referred to as an antigen. So type O blood, no antigens. Type O blood, though, has 2 antibodies. It has A antibodies and it has B antibodies. So if you want to start to create a little chart, that might be helpful, especially if you don't have a lot of background in the blood types. So just create a chart for the 4 types of blood, A, B, O, and AB. And then make 2 columns, one for the antigens and one for the antibodies. You might even want to make a third one because we're going to get into Rh factor here in a little bit. So no antigens on type O, and both antibodies are present. The next blood type that we have is type A blood. So for this one, we have our A blood cell again. This time you'll notice that it has structures sticking off the surface of the cell. So these are physically attached. The antigens are going to be physically attached to the surface of the blood. And these are actually A antigens. And so that is why we refer to it as being A blood type, is because it's getting named for the antigens that are on the surface of the cell. You'll notice that we still have antibodies here, but we don't have 2 different types of antibodies. This time we only see 1. And your type A blood is going to carry the B antibody. That's very important because you'll see a pattern here in just a second. So we have our A blood cell, has A antigens on the surface, and has B antibodies floating around it. I also want you to pay attention to the shape of the antigens because that is a very important feature as well. So the next one that we have is going to be our type B blood. And so we have our type B blood cell. Again, we see those structures on the surface. So those are our antigens, but they look different than the antigens for our type A blood cell. So this time we have B antigens on our B blood cell. That makes sense. And if we look at the antibodies that are associated with that, we have A type antibodies. So it's an inverse relationship. Whatever type of antigen it has the opposing or the opposite antibody floating around it. And then the last one that we have is going to be our AB blood. So we see our AB blood cell. And this time you'll notice that we have structures on the surface of the cell, but we have both types of antigens. We have A and B antigens on the surface of the cell. And if you look at this particular card, we have no antibodies. So again, for all 4, you have that inverse relationship. Whatever antigen is present, you have the opposing antibody that is going to be there. So what does this mean for doing blood transfusions? Because when they first started doing blood transfusions, they would take person A And they would be donating the blood, they would hook them up to person B, they would do the transfusion, both people would get up, walk away, and everything was a success. And then they would repeat the same process. They wouldn't change anything but the people. And all of a sudden, in a matter of minutes, the person giving the blood was fine, but the person getting the blood would end up dead. And they couldn't figure out why it was successful in some people and it was killing other people. And it took until they learned about the presence of the antigens and the antibodies to understand why some are successful and others are not. So when you take a blood type and you introduce it to another blood type, what has to happen is that blood type that is receiving the blood has to be able to recognize the antibodies that are coming in. And if it doesn't recognize the antibodies, it's going to grab onto them. So when we do blood transfusions and we look at our type A and our type B person. So if I take my type B blood here and I introduce it to a type A person, so this is my donor and this is my receiver. Now, what's going to happen is when I introduce this blood into my type A person, it's going to see those A antibodies. And if you notice, now that shape feature becomes very important because those A antibodies are now going to fit into that space right there, and they're going to get stuck together. And when you start taking those blood cells and gluing them all together, that process is called agglutination. It takes the blood from being a liquid and makes it almost like a gel. And it gets so thick and so heavy that the heart cannot push it through the blood vessels. And the heart is working so hard to move that blood forward that it eventually can't take the force anymore and the person will end up dying. So when you're doing blood transfusions, it's very important for the person receiving the blood to be able to recognize what is coming into it. And if it doesn't recognize it and it sees it as a foreign body, it will latch onto it and gum up the blood, so to speak, and almost turn it into a jello-like consistency. We will talk more about that when we are face-to-face next time. The last slide that you have talks about the RH factor, and the RH factor, it doesn't show up on any of the cards that I had, but it works on the same idea. So RH factor actually comes because they first discovered this in a species of monkey, which was known as the rhesus monkey, which is spelled R-H-E-S-U-S. So they just took the first two letters of the rhesus monkey name and referred to it as the RH factor. The RH factor is simply telling you that your blood has a protein or it doesn't have a protein. There is no health benefit. There is no problems or issues if you have the protein or you lack the protein. It's like your hair color. Some people have blonde hair, some people have black hair. There's no advantage either way to that. So it's simply you have it or you don't. But it also is going to be considered when you do blood transfusions because, again, your body has to be able to recognize what is coming in. And if it doesn't recognize it, it will attack it and try to get rid of it. So if you have the RH factor, you are called RH positive. So when somebody says I'm A positive, they're telling you that they have A blood type and they also have the RH factor or they have that protein. If you don't have the protein, you are referred to as being negative. And so B negative blood, they have the B blood type and they are lacking, they do not have that particular protein. So when you're doing blood exchanges between people, you have to understand how that works. If you have somebody that is negative, they don't have that protein, you can't take somebody who's positive for the protein and donate to that negative person because that negative person is not going to recognize the presence of that protein. They see it as a foreign body, and again, they'll gum up the blood. So in the case of RH factor, if you are negative, you can only receive from another person who has or who is negative as well. If you are positive, you can receive from another Rh positive person. You can also receive from somebody who is Rh negative because they don't have that protein, it's not present. there's nothing there that you wouldn't recognize, so it wouldn't cause any problems. Now, I am going to go a little bit more into this when we meet face to face. There's a little bit more that I want to talk about. So if you're not fully comfortable with blood exchanges, that is perfectly fine. What I would like you to do between now and our next meeting, so you have the video to watch. I also gave you some label sheets for the heart. There were two in color and then one that was in black and white. If you would please fill those out and make sure that you bring those back with you to class next week. And then the other thing is I would like to do an activity that involves computers. So if you have access to bring a computer with you, please do. If you're comfortable working with somebody else, if you don't have a computer that you can bring, I will let you double up, no more than 2 people on any one computer, but if you have your own computer, just to keep people spaced out and things like that, please bring that with you to class next time we meet on Thursday. If you have any questions between now and our next meeting, please send me an email and I will talk to you next time we meet.